Good morning. So the challenge of doing combination therapy and doing it in high throughput screening with an unrational, I wouldn't say irrational, but a brute force approach is that the success rate of combination therapy tends to be from those screens a fraction of a percent. And even with a few hundred drugs, you will have hundreds of thousands, millions sometimes combinations that you can test, and the material that you have that you can test this with is limited. So the question is, can we come up with a rational way to prioritize which drugs are more likely to work with a given patient, especially combinations? When you go to the triple therapy idea, it becomes even more complicated. So we approach this from a different standpoint, and we ask, can we take cohort patient data, cross-sectional data, so one biopsy from each patient, and then from that data mine information that can tell us how the patients have unique disease-perturbed networks that are matched to their molecular characteristics. And the idea would be the following. So given a large enough cohort of patients, you would expect to have subgroups of patients that share the same molecular mechanisms that have gone awry and are responsible for the disease. And the challenge is to figure out who those patients are and then rank out of them based on the etiology of the disease and separate them from patients who have a completely different kind of mechanistic dysfunction. Now, the challenge, as Lee mentioned, is that this is a network problem where you can have a defect in one or a few genes, but the effect can reverberate across the entire network. And you run into the challenge of figuring out what correlates are also mechanistically correlated with the etiology of the disease and are causal and not just the consequence of the perturbation of the disease. And when you have high dimensional data from patients and you add mutations and gene expression profiles, methylation profiles, when you have a high dimension data set, the chance of finding correlations is very high. And the idea generally is that you look for, in the correlation-based approach, is you look for a change in one feature and see how it correlates with another feature. And the feature in this case that we are interested in is the etiology of the disease or drug responsiveness. And the chance of finding spurious correlations is very high, and there's a whole website devoted to finding spurious correlations that have no mechanistic underpinnings whatsoever with the effect that you're observing. And this is going to be a bigger and bigger challenge as we are able to generate larger amounts of data from patients. So we need a thoughtful approach that has underpinnings in known biological principles and mechanism, so you can constrain the search space using principles that will allow you to then make actionable predictions, meaning you can go in and perturb the network and get predictable outcomes. And the way we did this was by developing this algorithm, which is able to bootstrap its way through this high dimensional data set and pick features that are correlated. And correlation across the features is supported by shared mechanistic principles. For example, if you have two genes that are changing in a similar fashion across patients, then you would ask, do those genes also share some mechanism of regulation that we can discover to support that this correlation is not coincidental and is likely mediated by a common regulatory mechanism. And we can do that and ask, who are the subsets of patients who show this pattern and both have the pattern and have the mechanistic evidence for sharing the pattern. So what this algorithm does, and this is a movie that you'll see that the patients here, these are the genes in the patients, are being clustered. Patients are being added and removed into that cluster, and all the patients to the left of that vertical dashed line are patients that share a pattern and are separated from patients to the right of the dashed line, vertical line, who do not have the pattern. Now, these genes that are correlated in these patients also share sequence features in the genome. And you can use mutation data, methylation data, physical interaction data, et cetera, to then independently cross-validate whether there's evidence in other independent data sets that tell you that these are not spurious correlations, that these are mechanistically based. And here's an actual profile that we were able to pull out of a data set of 422 glioblastoma patients. And we found hundreds of, actually 1,000 signatures, of which 500 were disease relevant. This is one of those signatures. And in this signature, you have every dot on this plot here is a patient. And it's a measurement of 16 genes in that patient and how they change relative to some standard reference that we created from the entire data set. And we then rank ordered the patients based on the level of expression of these 16 genes. And we get this trajectory, and we also highlighted onto this the known subtypes, and Eric mentioned some, but clearly there are many more that we don't know about. So to some extent, we can see that the mesenchymal subtype is enriched to the right side of the trajectory here, all the orange marked patient points here. And they're separated from the other subtypes. But you can see that the stratification of patients along the trajectory is not clean. So you can imply from that either the signature is incorrect, is noisy, or that the diagnosis of the patients and the subtyping was erroneous. There's an independent way you can start validating these kinds of predictions. You can take clinical outcomes, responsiveness being one to drugs, or survival being another, and something Eric also showed earlier. So we can ask, if you take patients from the lower quintile and the upper quintile and look for clinical outcomes such as survival, do we see a clear difference? And this information was not used to discover this trajectory. And you find that you can find evidence that the clinical outcomes of the patients that are grouped in the different parts of the trajectory are quite different. Now, the kind of data that Parvi mentioned in terms of drug responsiveness, if that were available for this trajectory, then we can look at the patients within each quintile group and ask, how did they respond to the different drugs, at least in the high throughput screening that was done? And is there evidence that there's a signature that is beautifully stratifying patients based on their responsiveness to one or more drugs? Then we can dive in and ask, what is the mechanistic basis for this signature? And there's a whole slew of ways we can decipher the networks, and I won't go through all of these, but they essentially bring in different algorithms to link mechanistic basis for the reason the genes are behaving in the way they do across those patients. And I'll give you a couple of examples here. So one is figuring out the transcriptional network that is perturbed within these patients. And we can take data from efforts such as ENCODE, which is a large consortium that is funded by NIH, and they have been mapping accessibility of chromatin across many of the human cell types. And the technology essentially asks, where in the genome of a given cell type can the enzyme DNase 1 go and cleave the DNA? And based on that information, you can create maps of accessibility. So you can say this region of the chromatin was accessible and open, so most likely functional in expressing genes, whereas this region was not accessible, and so probably not active. And we can then take known information regarding binding sequence, recognition sequence of transcription factors, there's thousands of those, and we can map it through those accessibility maps. And we can see, for example, there's this regulator called USF1 that has 3 possible locations that it could bind in this example. And of the 3 locations, only 2 locations were accessible to the enzyme. So the chromatin was open, suggesting that it's likely that the transcription factor could go and interact with this location in the genome. But then we can go even deeper at higher resolution, at nucleotide level resolution, and ask, is there evidence that there was a footprint? Meaning if the protein was sitting at that location, it would block accessibility of the enzyme at the very specific location, even if the chromatin was open. And so in the second plot here, over here, you see that the region was open and blocked at that very specific location where the transcription factor might have been bound. And so among the 3 sites, only 1 site was accessible and bound by that transcription factor. And we created a large database of such interaction maps for all of the known human transcription factors for which recognition sequences are known. And then we can map this onto the signatures that were discovered from the glioblastoma data set. We can do something similar for microRNA regulation. So these are small RNA molecules that can go and bind to the 3' untranslated region of transcripts. And we can ask genes that showed a similar pattern, did they also share a conserved short 6 to 8 nucleotide sequence in the 3' UTR in the transcript? And if they did, we can then hypothesize that a small microRNA molecule must have been responsible for interacting with those sites and then controlling the regulation of the genes. But we want to know which microRNA it might be. So it can take the sequence that is conserved and calculate the frequency of conservation at every position and convert that to a hidden Markov model and take the 1,000-odd known seed sequences for human microRNAs and then pass it through that hidden Markov model and identify the specific microRNA that must have bound to that location. So we can get down to very detailed mechanisms of why these signatures are perturbed in the patients. Now, these are testable hypotheses. And we integrated all of these different capabilities and more to create this pipeline. I won't go into any detail here, but it suffices to say that this creates a flow of information that links somatic mutations in patients and explains how those mutations perturb specific regulators in the network that act on these signatures that I described. And we can associate those signatures to clinical outcomes like survival and responsiveness and so on. So we have a map from mutations going down to a clinical phenotype. So this flow of information for glioblastoma identified about 34 mutations, somatic mutations, and also 68 mutations in pathways or networks that mapped onto these specific regulatory systems, and then explain how 500 signatures that stratified patients based on the different clinical phenotypes were affecting different hallmarks of cancers. And the network looks, so that's the key for this network. And the actual network looks, this is not working here, like this. It's a lot of information. I don't expect you to go through the whole map, but it shows you that from the tens of thousands of human genes, we have been able to pull out the specific networks that are perturbed across patients, and we can explain how mutations act on specific genes that are regulators that then control sets of genes across the entire human chromosome and affect clinical phenotypes such as hallmarks of cancers. And we can find all kinds of hypotheses about signature that is associated with invasiveness. For example, the one on the bottom left here can be mapped to specific somatic mutations that act in two genes, NF1 and PIK3CA. And those mutations are predicted by the network to perturb the expression of IRF1, which is then predicted by the physical interaction map to be influencing a set of genes in a signature that is associated with invasiveness. Now, this is a prediction. Now, we can go into another data set and ask if you look at the invasiveness phenotype of this patient's tumor cells, is there evidence that all the patients who have mutations in this network have increased invasiveness compared to patients who do not have these mutations? And the answer is yes. You get a very significant difference in how their invasiveness phenotype appears. So there's hundreds, thousands of such predictions we can make from the network. And the neat thing is we know how to test it. We can take the patient's stem cells and look for a very specific phenotype. And we then worked with Patrick Patterson at the Fred Hutch Cancer Center to do a unbiased validation where what he did was he used a CRISPR-Cas9, which is this hot new technology to make targeted chromosomal lesions. And he knocked out all of the known 1,000 human transcription factors, pulled them together in patient-derived glioma cell lines and in normal neuronal stem cell lines, and asked if we were to now proliferate these cells, could we find perturbed behaviors when the specific transcription factor associated with glioblastoma is deleted? And then we asked, did our network make a good, accurate prediction? We did not use that data, but independently, were we right? And the answer is we were quite right. I mean, there was 26 very specific glioblastoma-specific transcription factors that we predicted would be associated with the proliferation phenotype. And if you knock those out, you would see a phenotype. And we did. We also did targeted validation. So we went and we blocked specific transcription factors, specific microRNAs, and we looked at phenotypes such as apoptosis and proliferation, and again checked to see if we were able to find perturbed phenotypes when we blocked these known, these predictive regulators. And that too validated a number of predictions. So we were finding a lot of new biology, a number of new driver mutations, and being able to figure out strategies to identify the molecular features that are mechanistically associated with the etiology of the disease. And this is what we can do now. So the data set that is being generated by Charles and Parvi is essentially giving you information on responsiveness of drugs uh, in stem cells from patients. And we want to know what's the mechanistic reason why they're responding differently. And they've taken transcriptome measurements of these patients. We can take those transcriptome measurements and map the patients onto the trajectory and then find which of the 500 trajectories segregates them from the patients that responded differently, and map those signatures back onto the network and find a specific subnetworks that are most likely responsible for the difference in response to drugs, and use that as a framework to then make predictions of combinatorial interventions. So you could find that the patient is responding to 2 or more drugs, and we can see whether those drugs map into the same network or complementary networks, and whether we can combine them. In fact, I would argue that you might see that the patients did not respond to some drugs individually, but we can predict that if you actually combine this drug with a second drug, you would get a synergistic effect. And you could even potentially find cases where there was very little responsiveness to each of the individual drugs, but the network predicts that if you combine them, you'll get a very significant effect. So the single drug screens provide means to begin to understand and make predictions that we can then go back and test in those patient-derived cell lines. And so here's an example. So there's theory that if you target a gene at 2 different levels, one with a chemical, a drug, an established inhibitor or a new inhibitor that targets the protein product of that gene, and use a microRNA mimic that targets the transcript of the gene. And when you combine the two, the likelihood of finding synergistic effects is higher than random. And we found such an example with romidepsin, an established therapy which is highly toxic, has low half-life in blood, and we saw that it targets this gene. It is known to target the gene HDAC5, which falls into a signature that is predicted to be controlled by a microRNA. So we then made a prediction that if you combine the mimic that blocks the microRNA with romidepsin, you could lower the dosage of romidepsin but still have the same effect on apoptosis. And we confirmed that. So these, in fact, the IC50 profiles here for individual drugs are something that is already being generated in the high throughput screening, but we can add additional therapies or inhibitors that could be tested to generate the IC50 curves, and then we can do these 2x2 matrices of drugs, and in fact we can go to triple therapies as well quite quickly. And we see here, for example, romidepsin, when you add it in high dose, high strength, it has complete killing effect on the patient-derived stem cells. But as you lower the dosage of the drug, the killing effect goes away. You can recover that effect by titrating in the microRNA. So essentially, and all the cells in the heat map that have got blue borders in the cells are the ones where the effect was synergistic, meaning the dosage of the single agents did not have a significant effect on killing, but when combined with the second agent, the effect was much greater than you would have predicted over additive. So this is the strategy in words, and I can let you read it, but I can stop there, and I think we now move on to the panel discussion. So I can invite and I welcome questions at that point as well. Thank you.